So I want to just quickly go over what is due for the rest of the semester since we're getting toward the end. So we're in week 13 and we go to week 16. And I sent in an email yesterday, but just to really make sure everyone understands what's happening. So this week you actually have, oh, I'm sorry, we're on week 14. Gosh, sorry, behind a week. We're week 14. So we have 14, 15, 16. I was thinking 16 minus 3 is 13. 2 labs due this week, obviously the blood typing and the microscope lab. So if you're still having trouble with your brine shrimp, just find something else to look at, and that's totally fine. There, you can just find something in your yard that's a biological organism and look at it. Sounds like about 50% have hatched and 50% haven't. So I'm really sorry for those of you who didn't get your shrimp hatching. They're pretty cute, and it's a drag that they didn't work out so well. The blood typing lab, the part you submit are the questions. Some of you have already done that, but really to understand what to put in that table, you have to watch the video. So just really make sure you watch the video. And it ties in really well with what we're talking about this week, because antibodies are part of what's causing that blood typing lab to work. And we'll talk about that a little more today. The exam this week doesn't have any essay questions. It's all multiple choice, but the next one has 4. The last exam has 4 essay or short answer questions. So if you have questions about what this might be, you can feel free to come to my office hours and we can talk about it. Feel free to work ahead because, obviously that lab exam has 2 parts. It has a 25-point graphing assignment and it has a 25-point multiple choice assignment. So it's probably good to work ahead just so you don't have so much piled on you the very last week. Otherwise, you're going to have a 2-part lab exam and a lecture exam in the same week. But after this week, there are no more official lab activities. You'll just go back and review all of your labs since the last lab exam. So let's just quickly go through the list of what that includes. So lab exam 1 was in week 9. So it's everything after that. So that would include the enzyme lab. That would include, no, that was a 2-week lab. Forgot we extended that for 2 weeks. The mitosis and meiosis. So really knowing those images, being able to recognize interphase, prophase, metaphase, anaphase, telophase, visually recognizing those stages. So if I show you an image of that stage of mitosis, you would be able to name it. And then really for the microscope, I'll be honest, here are the things that I would ask you about the microscope. You need to understand that when you look through the ocular, the eyepiece, that is 10 times magnification on its own. And then each of those lenses has magnification. So if it's the 10x lens, that means the ocular is 10 and the lens is 10. That would be 100 times total magnification. So understanding those lenses and that, when you look through the eyepiece, it's going to multiply everything by 10. And then the prepared slides that you all looked at, I will have an image of one of those and ask you what it is. So that just requires students to have really looked at the slides and have their pictures of, what they actually looked at, so you recognize those. And then the blood typing lab, understanding how that works and being able to recognize a blood type. So if I had an image of a blood typing tray, being able to correctly name that blood type. So not a whole lot of material on that. And because I didn't do graphing on the last exam, we're doing graphing for this one. And again, that is a separate 25-point assignment. And some students have already done that and turned it in. So I think it's pretty straightforward. Any questions at all about the lab exam? It's 50 points. I think it's pretty easy. So, you should be able to get a pretty good score on that, hopefully. And so again, the only other lecture that is not currently posted is I am going to give a lecture on transmission of COVID. So next week, we start looking at bacteria and viruses and looking at different kinds of virus. There is a video on coronaviruses that I recorded last semester. And so I'm just going to add a little bit more. Here's one about COVID specifically, but I'm going to update that probably with some additional information, because obviously this has been evolving in what we know. And we do know a lot now about how it's transmitted. Okay, so let me stop sharing that, and I'm going to start sharing from here. Oops. Okay, so I'm just going to talk to you a little bit at the beginning about the immune system and how our immune system works. This does not in any way replace the videos on the immune system. Those are far more detailed than what I'm going to tell you today. But I just have to give you a little bit of an introduction before we start talking specifically about antibodies, how we get antibodies, and how vaccinations play a role in what we call our immune memory. And then specifically the different kinds of vaccinations that we have. Sorry, my Apple Pencil's on the floor. I have to get it. Okay, so again, this is just going to be a quick overview. It doesn't in any way replace the videos, but I just really want to bring everybody up to the same point if you haven't watched those videos yet. So you're born with some basic immune responses that we usually refer to as being general. Meaning they're non-specific. So it's the same response no matter what the pathogen or foreign particle is. And we call these also innate immune responses. So again, this is the type of immunity that you're born with. You don't acquire it over time. And it's really basic stuff, that it's the same response no matter what the foreign particle or pathogen is. And it really starts with your barrier defenses. And I talk a lot about barrier defenses in the immune system videos, but these are things that we just take for granted that keep pathogens out. So it's things like your skin. Your skin is a physical barrier. It's also a chemical barrier because your skin is slightly acidic, it's salty, you have sweat, oils, all these antimicrobial features of your skin. So that's why you go to bed at night and you don't wake up covered in mold every morning. And you know, I've talked a little bit in the video about when your skin is compromised, you know, if you have a cut or wound or a burn, that's when pathogens can get in because you've lost that physical barrier and that chemical barrier. Your stomach acid is a huge barrier defense. We eat a lot of things that we shouldn't be eating in our food, and stomach acid kills most of that. It's pretty rare that something slips through and makes us sick. It's all of the antimicrobial agents that are in tears, saliva, and mucus. Mucus lines all of your tracts. So any way something could get in, your respiratory tract, your digestive tract, urinary tract, reproductive tract, those all have mucus throughout, and that mucus has antimicrobial agents in it. And we also have hairs that trap things. So you have a lot of hairs throughout your respiratory tract that really trap things. So, you know, most of the time we keep the bad guys out. The bad guys aren't very easily able to get in, but they do, okay, because we do things to compromise our barrier defenses. And when that happens, we have a second line of general defense, and that is our inflammatory response. Our inflammatory response is very general. And when we say we're, quote, sick and we're feeling sick, we're really feeling the results of this inflammatory response. It's very general. It's the same really no matter what the pathogen is. And there are a lot of chemicals that are released by your immune cells to cause this response. It's how your white blood cells communicate with each other. So your white blood cells are a huge part of your immune system. And those white blood cells communicate with each other with chemical messengers called cytokines. And you might have heard this term associated with COVID infections, a cytokine storm. It's this inflammatory response that can kill you. So when you die from a viral infection, you're not dying because the virus released some harmful toxin into your body. You are dying because of your immune system's response to that virus. And it is the inflammatory response that's doing that. The whole idea is send a lot of fluid and blood to this area that includes a lot of white blood cells that are going to hopefully wipe out that invader, and a lot of fluid to flush that out. Increase body temperature. So when we run fever, it's part of our inflammatory response. You know, you get this on a small level if you've ever had an allergic reaction to something, you know, even just something like cat hair or pollen or dust, where you get, you know, runny nose, runny eyes get red. If you have a splinter and it swells up a little bit and gets red, that's an inflammatory response. When you get a cold, all that fluid that's in your lungs, same with the flu, that's an inflammatory response. So depending on your immune system's reaction to that pathogen, that inflammatory response can be pretty big. And that's what killed people in the 1918 flu pandemic. Is the youngest, healthiest immune systems had the largest inflammatory response to that pathogen. And unfortunately, because that infection was in the lower lungs rather than in the upper respiratory tract, those people drown in their own fluids. And they suffocated from that inflammatory response. Eventually, if the immune system realizes this inflammatory response is not enough, you know, sending all these white blood cells in to destroy the pathogen, raising body temperature, you know, as you know, raising body temperature, that denatures proteins, right? So remember, proteins function in a narrow range of pH and temperature. So increasing temperature is your body's way of denaturing the enzymes of that pathogen, but of course it also affects your own enzymes. So running fever for a long period of time and running too high of a fever is dangerous because it's affecting your own enzymes too, but it's also affecting the replication of that pathogen, either bacteria, virus, fungi, whatever the pathogen is. But over time, this eventually, if this isn't enough, it leads to what we call our specific or acquired immune responses. And that's where antibodies come in. I'm just going to quickly erase this and stay on the same slide. So our specific or acquired immunity kicks in over time if this inflammatory response isn't enough. So antibodies are really the ultimate goal of our specific immune response, because this is equivalent to what we call our immune memory. Oops, immune memory. Once you produce antibodies in response to a foreign pathogen or any type of foreign particle that gets into your body, some of those antibodies stick around. Okay, so that doesn't mean that that pathogen can't get in anymore. There's not this magical wall up. So if you have been vaccinated or you have some antibodies now because you were infected with COVID-19, it doesn't mean that COVID can't get into your cells anymore. It just means you're already armed and ready when it does, and you won't go through this long inflammatory response and get sick. that inflammatory response can last, anywhere from 10 to 14 days before antibodies are produced. And unfortunately, that's when people can die. People can die during that 10 to 14 days because of that strong inflammatory response, making their body sick, weak, everything it does to your body during that time before antibodies are produced. Once you get antibodies, antibodies are very specific. Okay, there are no general antibodies. They are very specific to that pathogen, and they bind to that pathogen and keep it from getting into your cells. They're binding to a very specific region or protein on that pathogen to keep it from getting into your cells. And those look like this. So I talk about antibodies in the general videos on the immune system, but I just really want to talk about them again, because what vaccinations do is cause you to produce antibodies without you getting super sick. So the two ways we can get antibodies are through natural infection or exposure, or artificially through vaccination. So this is an antibody, and you can see this term antigen. So an antigen is any foreign particle that elicits an immune response and can eventually result in you producing antibodies. And only one specific shape of antigen will fit into that receptor, that binding site on the antibody. But once you have those antibodies, some of those stick around. And if you're ever exposed again to the real thing for a second time, or the real thing if you've been vaccinated, you already have some antibodies there, and they start reproducing immediately upon exposure, and you have this whole army of soldiers ready to fight that pathogen before you ever get sick. You will not get that full-blown inflammatory response. Any questions about that before we keep rolling here? So this is just repeating what I just said. There are really, two ways that you can get antibodies. One is through natural infection or exposure. Sometimes you'll develop antibodies without having ever really been, quote, infected. And we're learning more about tuberculosis in particular in this way. We do a lot of TB testing in California. By the way, it's really interesting. If you look at the stats, they say that we have the highest rate of tuberculosis infection in California, but it's because we do the most testing. So if you do the most testing, you're going to have more positive cases, right? But a lot of people have never had tuberculosis, but they're just either, they're peripherally exposed without ever getting sick. And in fact, if you're from certain parts of the world, you're like probably 98% going to test positive for TB, even though you never actually had tuberculosis. So sometimes just exposure to the pathogen will cause you to produce antibodies. And then obviously artificially acquired through vaccination. This next slide just kind of gives you an overview. You don't need to know these details, but a lot of people ask me, how do antibodies work? Like, what are they really doing? And you have a good example of that this week. So in the blood typing lab, you are adding antibodies against 3 different antigens, 3 different markers that can be on your blood cells. And if that marker is present, those antibodies will bind to the blood cell and cause what's called agglutination. And we, in our blood typing lab, we're calling that clumping. Agglutination is when the antibodies, which in that picture you can see are the little purple Y-shaped proteins, they actually bind to the pathogen or the foreign particle and keep it from being able to get into your cells. It's also part of presenting it to the immune system. So now these phagocytic white blood cells can do cellular eating. They can actually devour and destroy that pathogen because it's being presented to the immune system as, look, this is foreign, come gobble it up. So that's what's happening in the blood typing lab. There are also other ways that antibodies can neutralize or really just prevent those foreign particles from entering your cell. And they can also cause lysis of the pathogen, causing little holes to form, and then the pathogen just lyses or bursts. So again, you don't need to memorize anything about that. I just put that in like as a curiosity thing. These people often ask, how do antibodies actually work? Also, just want to quickly address this term, herd immunity, because we're hearing it a lot right now. And it's really when enough of a population is immune to a pathogen or infectious disease that it really protects the entire herd. Okay, it protects the entire population, even those who aren't vaccinated or who do not have antibodies. So this obviously could happen through natural infection, or it can happen through vaccination. If you're counting on natural infection to reach herd immunity, you're also counting on a huge percentage of your population dying. And we obviously don't want that happening in any population of organisms, probably, but especially not in humans. We don't want so many people getting infected that enough of the population dies, and now we have herd immunity. And that's why we have vaccinations. This next picture is a little confusing, but what it's showing you is in the first picture, all of the blue individuals are not immunized, but don't have the disorder. And now 2 members of the population get the disease, whatever that is. Let's just say that it's COVID-19 infection. So those 2 individuals in red get infected, and now they pretty much infect almost everybody. The second example, just the ones in yellow are immunized. Okay, so the rate of infection goes down a little bit, but really, if you look who's really protected, it's the yellow guys. The yellow guys here, they're still the ones that aren't getting infected, but pretty much everybody else got infected because we didn't reach herd immunity. That was not enough protected individuals. In this very last example, you can see that everybody in yellow is vaccinated. All of these blue guys are not, but that's enough people being vaccinated that even with these 2 people who are infected, only 1 additional person gets infected. Okay, and it was one of the blue guys. It was one of the people who weren't vaccinated. But we protected those other blue guys because we were all vaccinated. So that's what herd immunity is. And really, how many need to be vaccinated to reach herd immunity depends on how that disorder is transmitted. If it's airborne, like COVID-19 is, like measles, mumps, and rubella, measles in particular, measles is highly infectious, airborne. And so the threshold is pretty high for how many need to get vaccinated. We don't really know what it is right now for COVID-19. Fauci is saying 70%-ish, maybe even up to 80, but there are scientists who are saying like 85 to 90 because it is so infectious and contagious. It's so far more contagious than the flu. I wanted to show you this slide that's got some thresholds on it, and it's kind of hard to see this, but what's interesting about the flu is we only need like 33 to 44% of the population vaccinated to get herd immunity. So believe it or not, the flu is not nearly as contagious as COVID-19 is. So all of those people who are saying, it's just like the flu. Well, no, actually it's not, because we really need about 90% probably of the population vaccinated to really reach herd immunity, or people being naturally infected, which is obviously happening too. If you look at measles, it's way up there. We need like 92 to 95% vaccinated to get herd immunity for measles, because it is so infectious and it's airborne. So that's what they're guessing is probably the case with COVID. It's probably more in the high 80s or 90s. And so, hopefully over time, more people will feel comfortable and get vaccinated. And we'll talk about that today a little bit more. Vaccines have increased human lifespan by more than 30 years. And vaccines have eradicated certain diseases in the US completely, smallpox and polio being 2 good examples of that. And vaccines now prevent 18 communicable diseases that used to kill thousands and thousands of people. If you look at human life expectancy, it used to be around 30 years old. That was the average lifespan of humans for hundreds of years. The two reasons that human lifespan has increased so dramatically: antibiotics and vaccinations. Those are the two biggest reasons that it's now in the 80s rather than the 30s. That's a huge increase in human lifespan. So vaccinations are an incredibly important part of our healthcare in saving lives, especially in children. So if you look at child mortality rate, meaning kids who die before the age of 5, that used to be around 50% in the United States. 50% of kids used to die before the age of 5. Why? Communicable diseases and infections. They didn't have antibiotics, they didn't have vaccinations. Vaccinations have changed our world for the better. So, here we are in the midst of the biggest global pandemic since 1918. And for the first time in history, we have people questioning science. And that's very scary. And, it's a lot due to misinformation that's out there. I personally think the media plays a really big role in that, but it's also the lack of science education. Even within my own extended family, there are people who are not getting vaccinated, and they have so many misconceptions, and it doesn't matter what you say to them because they don't understand basic biology. I love that you all now have this foundation of knowledge, and you can start questioning things you hear in the news, and you can go, well, that doesn't make sense. In fact, I had a friend email me this morning and tell me, I really think the pH of my body is off. So she bought these expensive drinks to start drinking to get her pH back in balance. And I said, where do you think that goes when you drink it? When you drink that drink, where does that go? Well, to my stomach. Okay, what is the pH of your stomach? I don't know, it's pretty acidic. Yeah. So, just simple things like that. But when it comes to vaccinations and understanding what these mRNA vaccines are doing and what they're not doing, it's really important. So, I'm hoping that after this class, you all can spread the knowledge and share your scientific knowledge with others, because it's sorely lacking in our country right now. So let's start talking about the different kinds of vaccines, and then really look specifically about, what are these messenger RNA vaccines, and should we be concerned, and what are they really doing in our cells and in our bodies in general. So we have a lot of different kinds of vaccines that are out there, and it's hard to read this, so I'm going to kind of walk you through the details here. But we have what are called whole vaccines, meaning it's the whole virus or bacteria. And what they do is they either do a killed version, so it's completely inactivated, or it's what's called live attenuated, meaning it's still the living pathogen, but they've taken out the part that makes you sick. So they've modified that pathogen to the point where it's going to trick your immune system into thinking it's the real whole infectious organism, but they've taken out the parts that would normally make you sick. So what are some good examples of these whole vaccines? Well, a good example of the inactivated whole vaccine is polio. And I don't know if you have relatives who were around in the 1950s who can tell you what it was like when kids started dying of polio, becoming paralyzed, permanently disabled from polio. But when that polio vaccine came out, people were lining up for miles to get their children vaccinated. And there was no questioning of science. It was, this is going to save my child's life, or save my child from being permanently paralyzed or disabled. And they went to any length to get that vaccine for their children. Just a completely different mindset. And that's before scientific knowledge was as great as it is now. Live attenuated, good examples of this would be the MMR, which is the measles, mumps, and rubella, and the chicken pox. Those are both live attenuated, meaning it's the whole organism, and they've just slightly modified it so that it's weakened to the point where it's not going to actually make you sick. Of course, the whole idea is your immune system perceives this as foreign and produces antibodies. And those antibodies stick around. So if you're ever infected with the real thing, you're already armed and ready. You already have that army of antibodies ready to defend you without you going through the inflammatory response that could kill you. Okay, more recently, we started producing vaccines that are really only a part of that pathogen. Believe it or not, it takes a really long time to cultivate, to produce these whole vaccines. If you're having to replicate viruses or bacteria and then modify them to the point where they're not going to make somebody sick, and imagine how many millions of doses you might need, that takes a really long time. And so we've found new and better ways of doing that are faster and more efficient and actually produce even more of an immune response in terms of antibody production. And that is just using parts of the organism, and those are called component vaccines. And you've already had component vaccines as part of the vaccine arsenal for a while. Protein subunits, so just taking whatever protein from that pathogen that it normally uses to infect your cells, and just inject just that protein into a person, and they will produce antibodies against that protein. And then when that protein is part of the whole virus or bacteria, the antibodies will still bind to it just the same. We've been getting pertussis as part of our Tdap or DTaP, depending on which one you got, vaccine for a while. This is also called whooping cough. And the pertussis part of that shot is just a protein subunit. One vaccine category that's not on this chart is what's called a toxoid vaccine. And tetanus is a good example of this. What they do is they inject an inactivated toxin that bacteria normally produces, and we produce antibodies against the toxin. So the reason is it's too hard to stop that bacteria from getting into your cells or into your bloodstream. So if we can produce antibodies against the toxin, even if we get infected with tetanus, we're now protected against the toxin that it releases. And hopefully you'll never get, lockjaw and all the horrible things that happen from tetanus. So I'm not going to go through all of these, but the next one I want to talk about is the one that affects us the most right now, and that is the RNA-based vaccines. So looking at the COVID vaccines, we have the Johnson & Johnson and the Pfizer and the Moderna. So the Johnson & Johnson, what they did is they actually took an adenovirus, which causes a common cold, is what we typically call what we get from the adenovirus. So this is a common cold virus. And what they did is they modified that virus. They added a gene for the COVID spike protein. Next week, you're going to start learning how viruses infect your cells. And what viruses do is, so virus is this very simple organism. Well, it's not even a living organism. It's this very simple particle, I guess you would call it. Has on the outside this protein coat. Oops. And then a very simple viral genome that can be either RNA or DNA. And then on the outside of that is this viral envelope that often contains some very specific proteins. And those proteins have a really specific shape. And typically we use a lock and key analogy. These would be the spike proteins. And if this virus, by good luck for the virus and bad luck for us, has a spike protein that perfectly fits into a receptor on one of your cell surfaces. So here's one of our cells, which would be obviously much larger than the virus in scale. But here's just a normal cell in your body. And this specific cell is an epithelial cell in your respiratory tract. And it has these receptors that are, they're there for specific molecules to bind to. And if that molecule binds, the cell knows that molecule belongs in here, wrap membrane around and bring it in. Well, these tricky viruses have co-evolved with our cells, and they just happen to have a spike protein that perfectly fits into that receptor. And so now they can bind, and guess what? Our cell readily brings them in because the key fits the lock. And once they're in, they inject their genome and some key enzymes, and now our cells become a reproductive factory. So what the Johnson & Johnson vaccine has done is it's taken the gene for that COVID spike protein and it's put it into the DNA of an adenovirus that really doesn't make you that sick, modified that virus to now contain the part of the COVID virus that we need to protect ourselves against. So we're producing antibodies against that spike protein. So now that those viruses can't get into our cell. Pfizer and Moderna have taken a completely different approach. And it's an approach that's been in research for over 30 years. We have been researching and testing mRNA vaccines for over 30 years. And the way the mRNA vaccines work is rather than putting the spike protein in, put in the messenger RNA sequence for the spike protein. So let's think about that for a second. Thinking about the lecture material on transcription and translation, we have DNA that codes for the messenger RNA. And remember, the messenger RNA that gets produced from the DNA is for just one gene. In other words, it's going to just produce one specific protein. We don't make messenger RNA for all of our DNA at once. We just make it for that one gene that we need to make a protein for. And then remember, that goes out into the cytoplasm, and on a ribosome, we get protein translation. In other words, assemble the protein that was coded for by the messenger RNA. So what the Pfizer and Moderna vaccines are doing is injecting messenger RNA that codes for the viral spike protein. And now we make the viral spike protein in our cells, because really, remember that analogy of in the castle, the scribe is throwing recipes out to the chefs, and whatever the recipe is, the chefs cook it. Whatever the recipe is, the chefs cook it. So in our cells, the chefs are the ribosomes, and they are cooking viral spike protein. Now, in response to that, we perceive that as foreign, and our immune system is going to produce antibodies against the viral spike protein. So why would we take this approach? Why would we just put in the mRNA and have our cells make the viral spike protein? Why wouldn't we just put in the viral spike protein? There are a few good reasons for this. One is that they found that in a lot of cases, when you just put in the viral spike protein as a component, our cells just break it down. They destroy it without making antibodies. Okay, the goal, this is the big goal, right? Without antibodies, there's no point. That's what we're trying to achieve. So you need a method that produces the most antibodies. And what they found is that with some viruses, if you just put in the viral spike protein, our cells just break it down. It doesn't stick around long enough to result in this robust antibody production. So what they found is that they can get the messenger RNA into the cell. Then we start making a lot of the viral spike protein, and we start making antibodies as we're making this viral spike protein. The other thing is that, manufacturing the viral spike protein takes some time. So, one problem was just breaking down that protein. The other was, how do we even manufacture that? That takes a lot of time. These messenger RNA vaccines, once you sequence that gene and you know the exact mRNA sequence you need, you can start putting together synthetic nucleotides and you can crank this out very quickly. If we were having to manufacture these vaccines and grow them, flu vaccine is a good example. They have to start making the flu vaccine over a year in advance. We didn't have that much time with COVID. Okay, we needed to do this very quickly. But the problem is they needed to develop a package to deliver this mRNA that our cells weren't going to just break down very quickly. And they ended up creating this lipid package that delivers the mRNA. So the only reason we haven't had a bunch of mRNA vaccines in the past is they've just been working on how do we get this into the cell without our cells breaking it down too quickly. And it was a packaging issue. And they had really just figured that out successfully. Luckily for the human population, we had figured that out right before the COVID-19 infection hit pandemic level. So we were in a really good position. We'd been doing 30 plus years of research and testing on mRNA vaccines. And we were trying to figure out how to package it. And we had just figured that out. And then the pandemic hit. So all that Pfizer and Moderna had to do is get that gene sequence from China and immediately start cranking out the vaccine. And within 2 weeks, they were able to get that vaccine into people in clinical trials. So the first people to receive the Pfizer and the Moderna vaccines were way back in February of 2020. And those people have now received their 1-year booster. Okay, so people from the original clinical trial, they got their first vaccine in like February, second one in March of 2020. They're now 1 year post second vaccine. They've now received a booster that includes a slight modification of the original plus the South African variant of the virus. The next problem with these vaccines was, how do you not cause too much of an inflammatory response, too much of a cytokine response to the vaccine? Like, you want to get robust antibody production, but you don't want to overdo it. And that was really part of the original clinical trial too. In fact, I recently read a blog from a guy that was part of the original clinical trial for Moderna. And they had what they called the high dose group, the medium, and the low dose. He was part of the high dose group, and he got very, very sick. His second vaccine, he actually was hospitalized because he had such a strong inflammatory response to that second vaccine. So what we've ended up getting is kind of the medium dose is what they ended up approving for the rest of us. But that was a tricky part too. But the great thing about these mRNA vaccines is now that they figured out how to package them, we can start solving a lot of problems. And one of the things that was already in clinical trials was a Zika virus vaccine. I don't know if you remember, Zika virus was in the news a lot a few years ago, causing serious birth defects in pregnant moms, affecting the unborn babies, transmitted by mosquitoes. It was originally just in like South and Central America, and then it came into Florida and was starting to spread to other warm areas of the US, and it was a pretty scary thing. Her herpes virus. They've been working on an mRNA vaccine for that. Rabies. It's really the future of flu vaccines and also part of the future of cancer treatment. There is an mRNA vaccine currently in clinical trials for melanoma. And what it does is it is replicating a protein on the surface of the melanoma cancer cells. And we actually then produce that same protein. And then in response, we produce antibodies against the protein on the cancer cells. And our antibodies go attack and bind to the cancer cells and present them to the immune system, and our immune system destroys them. So that's a pretty awesome thing. So mRNA vaccines are not new. They've been in clinical trials. It wasn't a question of, is the mRNA going to hurt us? It was, how do we package it so that we're not breaking it down before antibodies are produced? How do we get the dosage right so we're not causing too much of an inflammatory response in people? So why are people afraid of the mRNA vaccine? People think it was approved too quickly. Okay, well, let's talk about that for a minute. The FDA, the Food and Drug Administration, is the agency in the United States that approves all pharmaceutical products. So all drugs and all vaccines have to get approved through the FDA. And there's a very strict protocol in the United States of clinical trials in testing on humans to make sure there aren't any harmful side effects before these get approved. The line is long. Okay, if you're a pharmaceutical company wanting to get a new drug approved, unfortunately in the United States, it's a very lengthy process because there are so many drugs in production right now. So yes, it normally takes a long time to get a drug approved or a vaccine approved. If you have a global pandemic that's killing millions of people, you're moving to the front of the line. Okay, it's not that you're taking a shortcut in your clinical trials. It's not that you're taking a shortcut and not testing it as thoroughly as you should. You're moving to the front of the line, and you are going to start that process immediately and get through that process in the same amount of time as it would have been no matter where you were in the line. When it was your turn to go, that's how much time it would have taken. So these weren't rushed. They were moved to the front of the line. Okay, would you rather have, a drug that reduces the effects of gout moved to the front of the line, or would you rather have the COVID-19 vaccines moved to the front of the line? Like, come on. So there were no shortcuts taken. Let's also think about what these vaccines are doing. And should we be concerned that this is now in our cells? Could we find out years from now that everybody who got the mRNA vaccines, either the Pfizer or the Moderna, is now at risk of something bad happening? So let's draw a typical cell. Here's your nuclear envelope. It's protecting your DNA. It's controlling everything moving in and out of the nucleus of your cell where your DNA is. And now you have this little piece of messenger RNA in your cell that codes for viral spike protein. Is there any way for that to even get into the nucleus of your cell and affect your DNA? The answer is no. It's impossible. It's impossible for it to even get in to start with. But even if it did, you all now know that RNA and DNA are made from different nucleotides. There is no way for RNA to integrate with your DNA. There's no way for it to cause changes in your DNA. They're made from something completely different. There is no way for that mRNA to get in and change your DNA cause any type of DNA change, mutation, error. There's no way for it to be passed to your offspring. Okay, the only way it can be passed to offspring is if it can get in and modify the DNA of your eggs or sperm. And we've already seen that there's no way for it to change the DNA of any of your cells. It's definitely not going to change the DNA of your gametes for you to pass that on to children. Could it do something else damaging in the cell? Like what? There's nothing it could do. There's nothing it could do. And in fact, you know, they call it a self-destruct. But what's happening, remember, the whole problem was they had to try to figure out a way to package it so it wasn't immediately broken down by our cells, because you want it to lead to antibody production. So, but guess what? It's still getting destroyed by the cells. It's just luckily now happening after antibody production. So it's getting destroyed. It's getting broken down. So all that's left at the most are some synthetic nucleotides, and that really, you know, that's not doing anything to you. There's no way for that to do anything to you. So it, you know, this is getting broken down over time. It doesn't even exist. So if, let's say you got your second shot, you know, a month ago, there is no part of that vaccine even existing in your cells anymore. It's gone. All that's left is your immune memory, those antibodies. That is it. There is no way for that to cause long-term effects. Any side effects from vaccines happen almost immediately, but it's never after 1 to 2 weeks after getting the vaccine, because whatever it was that was injected in you breaks down, it's gone, your cells destroy it. Remember, your lysosomes are going through and breaking down and recycling everything that's not necessary in the cell. So you've broken those down, and all that's left is your immune memory. So if a vaccine is going to have a side effect, it happens immediately. There's no such thing as long-term side effects from vaccines. There's no such thing as that. So I think the question was, that's a great question, Cynthia, the difference in approval process between the FDA and the CDC. So the CDC is the Centers for Disease Control, and they have jurisdiction in, really, they oversee part of the clinical trial process. And what they issue is emergency approval. So the CDC got involved in releasing those drugs for emergency authorization, because the FDA does like permanent authorization. And so I think what's happening now is both Pfizer and Moderna have now applied for their permanent authorization rather than just their emergency authorization. So I think my understanding, and I could be totally wrong on that, but my understanding is the CDC was involved because it was an emergency authorization, and they have say in that emergency authorization process, as far as the timing and moving them to the front of the line for the clinical trial and deciding who gets to move to the front of the line. Because obviously, you know, I hate to say it, but obviously millions of lives were at stake, but there's also, you know, billions of dollars at stake. So there were like 30 or more companies who had a vaccine ready to go. And so the CDC had to play a big role in deciding who was allowed to move to the front of the line, like who's already shown enough progress and enough data that we can trust that these are the ones who should be given emergency authorization to move forward with clinical trials versus all of these others. Because Pfizer, Moderna, Johnson & Johnson, those were not the only ones trying to get to the finish line. And there were billions of dollars at stake for those companies. So that's a big decision. And so, you know, I think a lot of times with those types of decisions, there are two agencies involved as kind of an oversight, because you don't want corruption in one agency making multi-billion-dollar decisions that not only affect the health and life of others, but also money is a big part of all of that. That's a really great question. Any other questions about those mRNA vaccines? I just have one more vaccine story to tell you. I'm going to quickly grab my water. I forgot to bring it over to the computer. It's still next to my bed. Just a second. So there's really just one more topic I want to address related to vaccines. And, you know, in past semesters when COVID wasn't rearing its ugly head, the number one question that my students had was related to, you know, should you vaccinate your kids? And what is this whole autism link? Talk to me about that. So I want to just talk very briefly about that because there are specific, you know, regions of the US, usually the more affluent areas, where people are still hesitant and not vaccinating their children. And that's why we've had these crazy measles outbreaks and other things happening when people don't want to vaccinate their kids. And especially now that the number of childhood vaccinations has risen to this high level, you know, people are even more reluctant. They're like, do I really want to give all that to my new baby? But I just want to assure everyone that, you know, there's been extensive research and there is no link between autism and vaccines. Not the vaccine itself and not any of the ingredients within any of our vaccines. There was one specific ingredient that was being questioned called thimerosal. And there were some concerns about the mercury levels in the thimerosal possibly being toxic to children, but not causing autism. They've done extensive research and they've shown that there is absolutely no link between autism and vaccines, not any ingredient of the vaccine at all, and especially not the thimerosal. Thimerosal, it does not exist anymore in our vaccines, except for a small amount in certain flu vaccines. And there are alternatives. So if people want their kids to get the flu vaccine, there are non-thimerosal options, but it was completely removed from the MMR vaccine between 1999 and 2001. So it's been over 20 years since that's been used in childhood vaccines. Also, where this all started is there was a fraudulent study published by a team of scientists who had tremendous financial gain from this fraudulent publication because they were producing false data to help a group of parents sue a vaccine manufacturer in the UK, saying that the vaccinations cause their children to have autism. It was a group of 12 children, and the data was completely fabricated. And when it came out that this study was completely false, the publication retracted the article, and those scientists were found guilty of deliberate fraud, and that they had falsified data, and that there was actually no causal link between the MMR vaccine and autism. But unfortunately, this study was really damaging because, you know, it's horrible when something bad happens to a child and people look for reasons. And it just so happens that kids get vaccines when they're little, and that's when kids also start showing signs of autism. You know, we could just as likely link autism to riding in a car seat, because if you think about all the things that little kids do, they get vaccines, they ride in a car seat, they eat baby food, you know, they like, you can't say something is causing another thing just because they happened at the same time. The only way you can show that true cause and effect is through scientific testing. And there, believe me, this is such a big issue. There has been study after study after study after study. And if you're interested, you know, the CDC actually has a lot of information about these studies and what the results were. So anyone who has concerns can look at what the ingredients are of every vaccine, what research has been done, what publications there are, huge study groups, not 10 to 12 kids. We're talking like huge study groups to show that the vaccines are safe. So, it's far more harmful to have your kid get measles. Measles kills kids. And it's tremendous suffering that goes on as well. So that's the story with that. Are there any questions about anything? Questions about the lab? Questions about the test? Anything at all? Okay, I'll have office hours, just, normal office hours tomorrow morning and tomorrow afternoon, Thursday morning, Thursday afternoon. I can meet with you another time if those times don't work. So just let me know if there's anything you need. Enjoy the rest of your week. Just let me know if you have questions.
Okay, thank you.
Thank you.
Thank you.